Chapter 9 Biomolecules. There is a wide diversity of living organisms in our biosphere. Now, questions that arise in our mind is: are all living organisms made of the same chemicals, that is, elements and compounds? You have learned in chemistry how elemental analysis is performed. If we perform such as analysis on a plant tissue, animal tissue, or microbial paste, we obtain a list of elements like carbon, hydrogen, oxygen, and several other and their respective content per unit mass of a living tissue. If the mass analysis is performed on a species of earth crust as an example of non-living matter, we obtain a similar list. What are the difference between the two lists? In absolute term, no such differences could be made out. All the elements present in a sample of earth crust are also present in a sample of living tissue. However, a closer examination reveals that the relative abundance of carbon and hydrogen with respect to other elements is higher in any living organism than in earth crust. Table 9.1. Table 9.1 in comparisons of elements present in non-living and living matter. Elements: hydrogen, percentage weight of earth crust, human body. Hydrogen 0.14, 0.5, carbon 0.03, in human body 18.5, oxygen 46.6, human body 65.0, nitrogen very little 3.3, sulfur 0.03, 0.3, sodium 2.8, 0.2, calcium 3.6, 1.5, magnesium 2.1, 0.1, silicon 27.7, negligible. Adapted from CNRRAO, Understanding Chemistry, University Press, Hydrate. In higher classes, you will learn about how to analyze a living tissue samples and identify a particular organic compound. It will suffice to say here that one extract the compounds, then subjects the extract to various separation techniques till one has separated a compound from all other compounds. In other words, one isolated and purified a compound. Analytic technique, when applied to the compound, given as an idea of the molecular formula and the probable structures of the compound. All the carbon compounds that we get from living tissue can be called biomolecules. All the carbon compounds that we get from living tissue can be called biomolecules. However, living organisms have also got inorganic elements and compounds in them. How do we know this? A slightly different but destructive experiment has to be done. One: why a small amounts of living tissue, say a leaf or liver, and this is called wet and dry it. All the water evaporate, the remaining matter gives dry weight. Now if the tissue is fully burned, all the carbon compounds are oxidized to gases from CO2 water vapor and are removed. What is remaining is called ash. This ash contains inorganic elements like calcium, magnesium, etc. Inorganic compounds like sulfate, phosphate, etc. are also seen in the acid soluble fraction. Therefore, elemental analysis gives elemental composition of living tissue in the forms of hydrogen, oxygen, chlorine, carbon, etc. While analysis for compound gives an idea of the kind of organic. Figure 9.1. Figure 9.1. Figure 9.1. Where is the figure 9.1? Figure 9.1. Diagrammatic representation of small molecule weight organic compounds in living tissue. Sugar, carbohydrates, glucose, ribose, glycine, alanine, serine, amino acids. Okay, sugar, glucose, ribose, amino acid, glycine, alanine, serine. Fatty acid, palmitic acid, glycerol, triglycerol, and R3 are fatty acid. Phospholipid, lecithin, fat and oil, lipid, cholesterol, adenine, purine, adenosine, adenylic acid, uracil, adenine, nitrogen bases, pyrimidine, nucleoside, uridine, nucleotide. Okay. The chemical and physical properties of amino acids are essentially of the amino carboxyl and the R functional group. Based on number of amino and carboxyl group, there are acids, example glutamic acid, bases lysine and neutral valine amino acids. Similarly, there are aromatic amino acids, tyrosine, phenylalanine, tryptophan. A particular property of amino acid is the ionizable nature of NH2 and COOH group. Hence, in solution of different pH, the structures of amino acid changes. B is called a zwitteric form. A, C, B. Zwitterionic form. Lipids are generally water insoluble. They could be simply fatty acids. A fatty acid has a carboxylic group attached to a R group. The R group could be a methyl CH3 or ethyl C2H5 or higher number of CH2. Group 1 carbon to 19 carbon. For example, palmitic acids has 16 carbon including carboxyl carbon. Arachidonic acid has 20 carbon atoms including the carboxylic carbon. Fatty acids could be scattered without double bond or unsaturated with one or more C double bond. Another simple lipid is glycerol, which is trihydroxyl propane. Many lipids have both glycerol and fatty acids. Here the fatty acids are found esterified with glycerol. They can be then monoglyceride, diglyceride and triglyceride. There are also called fats and oils based on melting point. Oil have lower melting point, example gingerly oil, and hence remain as oil in winter. Can you identify a fat from the market? Some lipids have phosphorus and a phosphorylate organic compounds in them. These are phospholipids. They are found in cell membrane. Lecithin is one example. Some tissue, especially the neutral tissue, have lipids with more complex structures. Living organisms have a number of carbon compounds in which heterocyclic ring can be found. Some of these are nitrogen bases: adenine, guanine, cytosine, uracil, and thymine. When found attached to a sugar, they are called nucleosides. If a phosphate group is also found esterified to the sugar, they are called nucleotide. Adenosine, guanine, thymine, uridine, and cytidine are nucleosides. Adenylate acid, Thymidalic acid, guanylate acid, uridylic acids, and cytidilic acids are nucleotides. Nucleic acids like DNA and RNA consist of nucleotides only. DNA and RNA functions are genetic materials. Primary and secondary metabolites. The most exciting aspect of chemistry deals with isolating thousands of compounds, small and big, from living organisms, determining their structures and, if possible, synthesize them. If one were to make a list of biomolecules, such a list would have thousands of organic compounds, including amino acids, sugar, etc. For reasons that are given in section 9.10, we can call these biomolecules as metabolites. In animal tissue, one notices the presence of all such categories of compounds shown in figure 9.1. Okay. These are called primary metabolites. However, when one analyzes plant, fungal, and microbial cells, one would see thousands of compounds other than these called primary metabolites. Example: alkaloids, flavonoids, rubber, essential oil, antibiotics, colored pigments, scents, gums, species. These are called secondary metabolites. Table 9.3. Table 9.2 List of representative inorganic constituents of living tissues: sodium, potassium, calcium, magnesium, water, and compounds: NaCl, CaCO3, PO4 3 minus, SO4 2 minus. And table 9.3 Some secondary metabolite: pigments, carotenoid, anthocyanin, alkaloid, morphine, codeine, terpenoid, monoterpene, diterpene, essential oil, lemongrass oil, toxin, arbin, ricin, lecithin, concanavalin A, drugs, vinblastine, curcumin, etc., polymeric substances, rubber, gum, cellulose. While primary metabolites have identifiable functions and play known roles in normal physiological process, we do not at the moment understand the role of functions of all the secondary metabolites in host organism. However, many of them are useful to human welfare. Example: rubber, drugs, species, scents, and pigments. Some secondary metabolites have ecological importance. In the later chapters and years, you will learn more about this. Biomacromolecules. There is one feature common to all these compounds found in the acid soluble pool. They have molecular weight ranging from 18 to around 800 Dalton approximately. The acid insoluble fractions has only four types of organic compounds, that is protein, nucleic acid, polysaccharides, and lipids. These classes of compounds, with the exceptions of lipid, have molecular weight in the range of 10,000 Daltons and above. For this very reason, biomolecules, that is, chemical compounds found in living organisms, are of two types. One, those which have molecular weight less than 1,000 Dalton and are usually referred to as micromolecular molecules or simply biomolecules, while those which are found in the acid insoluble fractions are called macromolecules or biomacromolecules. The molecules in the insoluble fractions, with the exceptions of lipids, and are polymeric substances. Then why do lipids, whose molecular weight do not exceed 800 Dalton, come under acid insoluble fractions, that is, macromolecule fraction? Lipids are indeed small molecular weight compounds and are present not only as such but also arranged into structure like cell membrane and other membranes. When we grind a tissue, we are disturbing the cell structures. Cell membrane and other membranes are broken into pieces and forms vesicles which are not water soluble. Therefore, this membrane fragment in the form of vesicles get separated along with the acid insoluble pool, and hence in the macromolecular fraction lipids are not strictly macromolecules. The acid soluble pool represent roughly the cytoplasmic composition. The macromolecules from cytoplasms and organelles become the acid insoluble fraction. Together they represent the entire chemical composition. The macromolecules from cytoplasm and organelles become the acid insoluble fraction. Together they represent the entire chemical compositions of living tissue or organism. In summary, if we represent the chemical composition of living tissue from abundance points of view and arrange them class-wise, we observe that water is the most abundant chemical in living organism. Table 9.4. Table 9.4 Average compositions of cell compound: water percentage of the total cellular mass 17 to 90%, protein 10 to 15%, carbohydrate 3%, lipid 2%, nucleic acid 5 to 7%, iron 1%. Protein. Proteins are polypeptide. They are linear chains of amino acids linked by peptide bond, as shown in figure 9.3. Figure 9.3. Okay. Figure 9 point various levels of protein structure. Okay. Primary, secondary, tertiary, and quaternary. Each protein is polymer of amino acids, as there are 20 types of amino acids. Example: aniline, cytine, proline, tryptophan, lysine, etc. A protein is a heteropolymer and not a homopolymer. A homopolymer has only one type of monomeric representing n number of times. This information about the amino acids content is important, as later in your nutrients lesson you will learn that certain amino acids are essential for our health and they have to be supplied through our diet. Hence, dietary proteins are the source of essential amino acids. Therefore, amino acids can be essential amino acids. Therefore, amino acids can be essential or non-essential. The latter are those which our body can make, which while we get essential amino acids through our diet food. Proteins carry out many functions in living organisms. Some transport nutrients across cell membrane. Some fight infectious organisms, antibodies. Some are hormones. Some are enzymes. Exam table etc. Table 9.5. Table 9.5. Some proteins and their functions. Proteins: collagen function, intracellular ground substance, trypsin enzyme, insulin hormone, antibody fight infectious agents, receptors sensory reception, smell, taste, hormone, GLUT4 enable glucose transport into cell. Collagen is the most abundant protein in animal world, and ribulose biphosphate carboxylase oxygenase ribisco is the most abundant protein in whole the biosphere. Polysaccharide: the acid-insoluble pellet also has polysaccharide carbohydrates as another class of macromolecules. Polysaccharides are long chain of sugar. They are thread, literally a cotton thread, containing different monosaccharides as building blocks. For example, cellulose is a polymeric polysaccharide consists of only one type of monosaccharide, that is glucose. Cellulose is a homopolymer. Starch is a variant of this but present as a storehouse of energy in plant tissue. Animal have another variant called glycogen. Insulin is a polymer of fructose in a polysaccharide chain, say glycogen. The right end is called the reducing end and the left end is called the non-reducing end. It has branch as shown in the form of a cartoon figure 9.2. Figure 9.2. Diagrammatic representation of portion of glucose. Starch from helical secondary structures. In fact, starch can hold I2 molecules in the helical protein. The starch I2 is blue in color. Cellulose does not contain complex helicals and hence cannot hold. Plant cell walls are made up of cellulose. Paper made from plant pulp and cotton fiber is cellulosic. There are more complex polysaccharides in nature. They have as building blocks amino sugar and chemically modified sugar. Example: glucosamine, N-acetyl galactosamine, etc. Exoskeleton of arthropods, for example, have a complex polysaccharides called chitin. These complex polysaccharides are mostly homopolymer. Nucleic acid. The other type of macromolecule that one would find in the acid-insoluble fractions of any living tissue is the nucleic acid. These are polynucleotides together with polysaccharides and polypeptides. These comprise the true macromolecule fractions of any living tissue or cell. For nucleic acid, the building blocks is a nucleotide. A nucleotide has three chemically distinct compounds. One is heterocyclic compound, the second is monosaccharide, and third is polysaccharidic phosphoric acids or phosphate. As you notice in figure 9.1, the heterocyclic compound in nucleic acids are the nitrogenous bases named adenine, guanine, uracil, cytosine, and thymine. Adenine and guanine are substituted purine while the rest are substituted pyrimidine. The skeleton heterocyclic ring is called as purine and pyrimidine respectively. The sugar found in nucleotides in either ribose, a monosaccharide, pentose, or second deoxyribose. A nucleic acid containing deoxyribose is called deoxyribonucleic acid DNA, while that which contains ribose is called a ribonucleic acid RNA. Structure of proteins. Proteins, as mentioned earlier, are heteropolymers containing straight of amino acids. Structures of molecules means different things in different contexts. In inorganic chemistry, the structures invariably refer to the molecular formula, example NaCl, MgCl2, etc. Organic chemists always write a two-dimensional view of the molecules which representing the structures of the molecules, example benzene, naphthalene, etc. Physicists conjure up the three-dimensional views of molecular structures while biologists describe the protein structures at four level. The sequence of amino acid, that is the position information of protein, which is the first amino acids, while the secondary and as on is called the primary structures. Figure 9.3a of a protein. Protein is imagined as a line, the left end represented by the first amino acids and the right end representing by the last amino acid. The first amino acids is also called as N-terminal amino acids. The last amino acids is called C-terminal amino acids. A protein thread does not exist throughout as an extended rigid rod. The thread is folded in the form of helix, similar to a revolving staircase. Of course, only some portions of the protein thread are arranged in the form of a helix. In protein, only right-handed helix are observed. Other regions of the protein thread are folded into other forms in what is called the secondary structure, figure 9.3b. In addition, the long protein chain is also folded upon itself like a hollow woolen ball, giving rise to the tertiary structures. This gives us a three-dimensional view of a protein. Tertiary structures is absolutely necessary for the many biological activities of protein. Some proteins are an assembly of more than one polypeptide or subunit. The manner in which these individual folded polypeptides or subunits are arranged with respect to each other, example linear straight of pairs arranged one upon each other in the form of a cube or plate, etc., is the architectures of a protein, otherwise called the quaternary structures of a protein, figure 9.3d. Adult human hemoglobin consists of four subunits. Two of these are identical to each other, hence two subunits of alpha types and two subunits of beta types together constitute the human hemoglobin. Enzymes. Almost all enzymes are proteins. There are some nucleic acids that behave like enzymes. These are called ribozymes. One can depict an enzyme by a line diagram. An enzyme, like any protein, has a primary structure, that is amino acid sequence of a protein. An enzyme, like any protein, has the secondary and tertiary structures. When you look at the tertiary structure, figure 9.3d, you will notice that the backbone of the protein chain folds upon itself. The chain crisscross itself, and hence many crevices or pockets are made. One such pockets is the active sites, and active sites of an enzyme is a crevice or pockets into which the substrate fits. These enzymes, through their active sites, catalyze reactions at a high rate. Enzymes catalysts differ from inorganic catalysts in many ways, but one major difference needs mention. Inorganic catalysts work efficiently at high temperature and high pressure, while enzymes get damaged at high temperature, say above 40 degrees Celsius. However, enzymes isolate from organisms who normally live under extremely high temperature. Example: hot wind and sulfur springs are stable and retain their catalyst power even at high temperature upon 80 degrees Celsius to 90 degrees Celsius. Thermal stability is thus an important quality of such enzymes isolated from thermophilic organisms. Chemical reactions. How do we understand the enzymes? Let us first understand a chemical reaction. Chemical compounds undergo two types of changes. A physical change simply refers to a change in shape without breaking of bonds. This is a physical process. Another physical process is a change in state of matter. When ice melts into water, or when water becomes a vapor. These are also physical processes. However, when bonds are broken and new bonds are formed during transformation, this will be called a chemical reaction. For example, BaOH2 plus H2SO4, BaSO4 plus 2H2 is an inorganic chemical reaction. Similarly, hydrolysis of starch into glucose is an organic chemical reaction. State of a physical or chemical process refer to the amount of product formed per unit time. It can be expressed as rate is directly proportional to delta of pressure and time, inversely proportional to delta of time. Rate can also be called the velocity of the direction is specified. Rate of physical and chemical process are influenced by temperature among other factors. A general rule of thumb is that rate double or decreased by half for every 10 degrees Celsius change in either direction. Catalyzed reactions process at a rate vastly higher than that of uncatalyzed one. When enzymes catalyzed reactions are observed, the rate would be vastly higher than the same but uncatalyzed reactions. For example, carbon dioxide, water, carbon anhydrase, H2CO2, carbonic acid. In the absence of any enzyme, this reaction is very slow, while about downstream molecule of H2CO3 being formed in an hour. However, by using the enzyme present within the cytoplasm called carbonic anhydrase, the reaction spread dramatically with about 6 lakh molecules being formed up every second. The enzyme has accelerated the reaction rate by about 10 million times. The power of enzymes is incredible. Indeed, there are thousands of types of enzymes, each catalyzing a unique chemical or metabolic reaction. A multi-steps chemical reactions when each of the steps is catalyzed by the enzymes complex or different enzymes is called a metabolite pathway. For example, glucose to pyruvic acid is actually a metabolic pathway in which glucose becomes pyruvic acid through 10 different enzymes catalyzed metabolic reactions. When you study respiration in chapter 12, you will study this reaction. At this stage, you should know that this very metabolic pathway with one or two additional reactions gives rise to a variety of metabolic ends products in our skeletal muscles under anaerobic conditions. Lactic acid is formed under normal aerobic condition. Pyruvic acid is formed in yeast during fermentation. The same pathway leads to the production of ethanol, alcohol. Hence, in different conditions, different products are possible. How do enzymes bring about such high rate of chemical conversion? The understanding this, we should study enzymes a little more. We have already understood the idea of an active site. The chemical or metabolic conversion refer to a reaction. The chemical which is converted into a product is called a substrate. Hence, enzyme that is protein, which three-dimensional structure including an active site convert a substrate into a product P. Symbolically, this can be depicted as ESP. S is converted into P. It is now understood that the substrate S has to bind the enzymes at its active sites within a given cleft or pocket. This substrate has to diffuse towards the active site. There is thus an obligatory formation of an ES complex E state for enzymes. This complex formation is a transient phenomenon during the state where substrate is bound to the enzyme's active site. A new structure of the substrate called a transition state structure is formed very soon after the expected bond breaking making is completed. The product is released from the active site. In other words, the structure of substrate gets transformed into the structure of product. The pathway of this transformation must go through the so-called transition state structure. There could be many more altered structure states between the stable substrate and the product. Implied in this statement is the fact that all other intermediate structural states are unstable. Stability is something related to energy status of the molecule or structure. Hence, when we look at this pictorially through a graph, it looks like something as in figure 9.4. Concept of activation energy, progression of reaction, potential energy, substrate, activation energy with enzyme, activation energy without enzyme, transition state, product, transition state and product.